reactivation of p53 to prevent oral cancer. You see, p53, it's a phosphoprotein associated with tumor suppression. It does three things. Number one, if there is DNA damage, it can stop the cell cycle to prevent its replication. Number two, once it stops the cell cycle, can activate DNA repair enzymes. If for some reason it cannot repair the damage, it induces apoptosis. So as you can see, p53 doesn't allow damaged cells with damaged DNA going on and reproducing in the body. This is how p53 prevents tumors from appearing. If there is a mutant p53 not working properly, it is most likely to develop a tumor. Now, if there is a p53 that is actually not working properly, now what if there was a way to reactivate p53 and make it work properly again? Here is where hexylresorcinol goes in. Hexylresorcinol, also known from now on as 4-HR, is a chemical chaperone that restores function via acetylation and phosphorylation of p53. Oral carcinomas have a much higher index of p53 mutation. Mutant might hide the amino acids, not allowing to start apoptosis. Here is when chemical chaperone induces conformational changes that might expose these amino acids so they can get phosphorylated and acetylated. Here lies the secret of the anti-tumor effect. Cells identified as YD15 from tongue of patients diagnosed with mucoepidermoid carcinoma and YD9 cells from squamous cell carcinoma, derived from buccal chick. Both types of cells had mutant p53. They were grown in 6 well culture plates. Once grown to approximately 70%, they were treated with 1, 10, and 100 micrograms of 4-HR for about 2, 8, and 24 hours. Lysates were subjected to Western blotting to detect the expression levels of p53, p53, and ACP53. Proteins were collected after 2, 8, and 24 hours. Fluorescence intensity was measured with a plate reader. Results were outstanding. YD15 and YD9 cells with 4-HR resulted in a significantly increased expression of p53 and ACP53. As you can see, all of them showed increase, but treatments with 24 hours and 100 micrograms showed the best results. Fluorescence showed a green cytoplasm due to the release of the cytochrome C from mitochondria. Conclusion: 4-HR is a potential agent for recovering loss of function in mutant p53 and works as a pharmacological chaperone which induces phosphorylation of p53 and acetylation of p53. Therefore, it can be taken as a possible option on oral carcinoma preventive treatment.